And this is a fairly complicated process with lots and lots of steps. It takes a long time, but I can try to explain it in basically 3 basic steps. The first step is delivering the DNA to the plant or the plant cell. And we have a lot of ways that we can deliver the DNA and a lot of cells that we can deliver it to, but we found that one of the best types of cells to deliver the DNA to are embryos, which are baby plants that are inside the seeds of every seed. So here is, you'll surely recognize an ear of corn. And I'm going to pass this around in a minute. But this is an immature ear. It's not fully mature yet, doesn't have solid seeds. But you did this, right, Brooke? She cut the tops off of all of the seeds, the caps off the tops of the kernels, and inside those are the embryos. And I think you can even see one where it looks like one's been removed. So inside every kernel is an embryo. And then I'll pass this around to the other direction, and these are the actual embryos that are coming from an ear about that size. It turns out that if you deliver DNA to those kinds of cells and those kinds of embryos, it works really well. So we've been doing this for a long time. So that's what we deliver the DNA to. And how we deliver the DNA to it is by, have any of you heard of the gene gun?
Yes.
It seems like something everybody's heard about, and it was the way that we delivered genes to plants 20 or some years ago. You would actually coat your DNA onto little microscopic gold particles. You'd load them into a pressure device, which we call a gun. Doesn't actually look like a gun, but it'll actually shoot these little particles down into a Petri plate that has plant cells on it, like those that I'm passing around. And the little particles will collide with the cells, go into the cells, and since the DNA is on the particles, they'll go into the cells. Nowadays we use bacteria. So if you've ever been in the woods and you see a tree with a big blob growing out of the side of it, that's a plant disease called crown gall disease. And it's caused by a bacterium that naturally transfers DNA into cells. And what we and other scientists did about 30 years ago is figure out a way to engineer that bacterium so it'll deliver the genes that we want to into the cells. So when that DNA goes into the cells, let's pretend this is the DNA that we're transferring into the plant cells. And let's pretend that that big thing around the table is a corn chromosome. Now this is obviously not to scale. And if it were to scale, this thing would go down to Florida. That's how much DNA there is in a corn cell. So when we're transferring DNA into a plant cell, it's going to go into where the chromosomes are in the nucleus, and it will get inserted into the chromosome. So that's the gene inserting the chromosome. This gene could be a gene from BT. I don't know if you've ever heard of BT corn or BT cotton. So it's a gene that comes from a bacterium called Bacillus thuringiensis, and it'll make the plant resistant to insects. And that's a very common type of GMO that we've made and had a lot of success with.
Which is the one we showed you in the display cases.
Exactly,
So, yeah.
exactly. So I've just inserted a gene in there, so it's genetically modified. Remember, if this were to scale, this thing would go all the way down to Florida. So it's by comparison to what's already in there, it's pretty small actually. There's 30,000 genes in corn, and we've just added one more. All right, so that's the deliver part. We've delivered DNA to one cell on one of these embryos, these little embryos that you saw. There might be 50 or 80,000 cells on there, and now one of them has taken up the DNA. So it's pretty rare. So the next thing we have to do is find a way to make that cell and not all the other cells grow and divide and divide. And so we'll take those embryos and then put them onto other Petri plates, which I'll pass around, in a way that only the cells that have actually taken up the DNA can divide. And those.
Is there a difference other than the fact that you guys did it in a petri dish of splicing in the DNA like that, or like procreation?
Or procreation.
Like, having a child, right? So you're altering the two DNAs that are going together in that embryo, right? Is there really, like, the end result, is it really different doing it like this than having a child? Like,
So, so fundamentally it's the same thing. And what I just described is sometimes described as horizontal gene transfer. where genes will move from one species to another, and it seems very unnatural crossing species boundaries, but horizontal gene transfer has occurred throughout the history of life, basically. So already in the corn genome, there's lots and lots of genes in there from viruses and bacteria and so on.
Yeah, I guess I've heard some things out in the world, right? And so I get that we're talking about plants and they're plants, they're not humans, but I guess, like, how are we not genetically modified, right? In the sense of...
It depends on your definition of genetic modification.
Okay, I guess that's what I'm
getting at. Yeah. And as I said at the beginning, humans have been modifying genes without even knowing that there were such things as genes in DNA for tens of thousands of years. Every time they select the best seed out of a basket of seed and then plant that and do that over and over and over time the genes of that corn plant or that rice plant are definitely changing because of humans. But we understand that when people think about GMOs, they're actually thinking about something more like this.
Modern.
Even though, but it's a very good point that it's basically the same. So we're going to take that cell and we're going to select, we're going to put them on those kinds of Petri plates that are being passed around now, and only the cells that have successfully taken up the gene will divide and divide and divide. And that's something that we call Callus. It's a little greenish, but it doesn't look like a plant probably, but take my word for it, it's really plant cells, they're really alive, and they've got the gene in it. Now instead of one cell having the gene in it, now you've got a million cells that have got the gene in it. But that's Callus. And we sell seeds that come from plants. We don't sell callus. So then the third step in this process is we have to regenerate. So we'll take that kind of callus after we've done that selection step long enough, and then add different kinds of plant hormones. So plants have hormones just like animals, just like humans. And just like animals and humans, different kinds of hormones will cause different kinds of effects. Some hormones will cause the plants to start forming leaves, and other hormones will cause the plants to start forming roots. So as I pass these around, you'll see they have lots of leaves on them. These are corn plants. And then, especially on this one, if you look on the bottom of it, you'll see lots of roots starting to grow. And that's because we've taken this kind of callus, which doesn't have leaves and doesn't have roots, and start adding natural plant hormones to start inducing that.
Right, what kind of hormones do you want?
Yeah,
The ones that go straight to the bus.
So at this point, it may not look like it because they're in these flat Petri plates and they're kind of smashed down, so they may not look like plants, but they're really plants. And if we remove those covers, those leaves will stand up straight and it'll start to look like a baby corn plant. And in fact, at some point in this process, and this process will take 8 to 10 weeks. Yeah, so how long it takes, everything I'm talking about so far is corn, but the basic steps are the same for everything that we do, cotton and soybean and everything. So at some point, they'll stand up and they'll get a few inches tall and have 3 or 4 leaves on them. Can I open this? Or do you want to use it again?
We can use the other one.
Okay. So the problem is when we open these things, will get contaminated. I'm not going to open it. But in here, I'll pass this one around too. You'll see lots of roots on the bottom, and then you'll see the plants growing in there, and these are corn plants. And at this point, the making of the GMO is basically over. But it's the beginning of many, many years of testing. And one of the first things we're going to do is test it to make sure it has the gene that we want. So let's say we had a BT gene. We want to make sure it's there, that it's intact, that it's the way we want it to be. And so we can do that by taking just a little piece of the leaf off, less than a centimeter long, send it over to the labs. Did you guys see the chipper yet?
Not yet.
Okay, send it over to those labs there. They'll grind up that leaf, they'll extract the DNA, and within a couple of days they can do a series of basic tests on the DNA to make sure it is what we say it is and what we think it is. And that's still just the beginning. Now it's going to go for years and years and years on tests out in the fields, in the greenhouses, and so on. So that's basically how you make a GMO. You take some plant cells, you deliver some DNA to that cell, select that cell so it divides and divides and divides and forms a chunk of something we call callus, and then take that callus and regenerate a plant, and then start testing the plant. And eventually, you get a plant that produces its own ear, and a few hundred seed on it, and every one of those seed will have the gene in it. You plant those out, you get plants with seeds in it. Before you know it, from that one cell, Now you've got enough corn to cover the entire Midwest. To me, that's always amazing that it came from that one cell.
Are these specific to corn?